want them yourselves on YouTube for whatever reason, we don't have to upload them. So just, I guess the group would have to agree to that. And so Jenny, I'm sure you all know Jenny and I will be marking you up the back. But relax, have fun. I'm sure you've all worked really hard and prepared something wonderful. The talks are how long?
15 minutes.
Good. That's what they are. I was hoping that's what I said to you. So the talks are 15 minutes. We've got enough time to extend the questions out if you want, so don't rush through your answers. And I don't care whether one of you answers everything or you all answer the questions. And hopefully everyone else will be listening to the other talks and giving them positive affirmations from out in the audience, and also thinking of questions to ask them afterwards, after their talk. Thank you very much. Thanks. So we need, you're the director as well. So
No, step through. It's already recording.
All right. Hi, so I'm Julia. This is Callie. We've got Maya, Megan, and Daisy. And today we're going to be discussing a strategy to cure adult onset hyperlactasia using CRISPR-mediated base conversion. So this condition is also termed adult lactose intolerance, and it affects up to 75% of adults worldwide, wherein they are unable to effectively metabolize lactose. So the enzyme lactase fluorescein hydrolase, also simply termed lactase, is responsible for hydrolyzing lactose into glucose and galactose, which can then be absorbed across the intestinal epithelium. So lactase is found in the intestine of all mammals. And when lactose is not digested in the intestine, it then passes through into the colon, where it's fermented, and this can produce symptoms such as diarrhea, flatulence, and abdominal pain. So all humans are born with high levels of lactase expression, as it is required to break down lactose in milk, which is the primary nutrient for babies. So adult hyperlactasia is an autosomal recessive condition wherein lactase expression is downregulated after weaning, most likely due to a decrease in transcriptional levels. This is believed to be the ancestral phenotype, as we find this occurring in all other mammals apart from humans. So lactose intolerance is the predominant phenotype in many populations, including Asia, as well as native South American populations and some African populations. Though the age of the adult onset varies, so sometimes it's the ages of 2 or 3 onwards, sometimes it's found from teenagers onwards. So the mutant phenotype here is lactase persistence, in fact, which is an autosomal dominant condition where an individual is able to continue to express lactase throughout their adult life and hence break down lactose. This is believed to have arisen from a mutation 5,000 to 10,000 years ago. So the LCT gene, located on chromosome 2, codes for the lactase enzyme. 2 SNP polymorphisms associated with lactose intolerance have been identified, both located within introns of the MCM6 gene, which is located 3,000 base pairs upstream of the lactase gene. So a cytosine located at position 13910 has been found to have a 100% association with lactose intolerance, whilst those with lactase persistence contain a mutation allowing them to have a thymine in this location. So this region is believed to have enhancer activity, with the T variant enhancing lactase-promoted-driven reporter expression by about 40-fold, potentially due to a high recruiting of transcriptional activators more efficiently than the C13910 version. It is also possible that transcriptional repressors may be upregulated during lactase persistence. So today, whilst we will be focusing on this mutation in particular, it's also interesting to note that there is a second polymorphism at site 22018, which has also been located, wherein those with lactose intolerance contain a adenine, whilst those with lactose persistence contain a guanine. However, this region has no significant gene regulatory activity, and there's been no functional difference between the adenine and the guanine. And so now we'll just discuss how CRISPR works and how we're going to use this to solve this problem.
Alright, so we believe that the burgeoning field of base editing CRISPR technology is the key to curing adult onset hyperlactasia. So this technology was first derived from naturally occurring immune mechanisms in bacteria that have enzymes that are capable of finding and slicing through foreign nucleic acids such as virus-encoding DNA. And it was later discovered that these mechanisms had the ability to alter and modify DNA or RNA sequences, therefore giving rise to a new approach for precise and targeted genome editing. So CRISPR has two major functions, searching and cleaving. Firstly, it interacts with the internal mechanism and searches the genome to find a specific DNA or RNA sequence. And once a CRISPR-associated gene, such as Cas9, finds the target region, a double-stranded break is induced. So further editing can take place. However, before these two processes, the CRISPR-associated gene should already consist of a reference library, and this is created from fragments of phage genome from previous foreign invasions. And the invading DNA is fragmented, and these are the spatial regions shown here. So the invading DNA is fragmented next to a 3-base pair motif known as the PAM sequence, which is the protospacer adjacent motif. Hence, this means that DNA is not fragmented randomly, but it is in fact organized in a distinct sequence to form the reference library. So the whole organization allows for CRISPR-associated genes to recognize specific sequences of corresponding target DNA to carry out editing. So firstly, the CRISPR-associated gene, which is Cas9 in this case, forms a complex with a single guide RNA, shown here in red. So the single guide RNA then guides the Cas9 enzyme to a target region, shown over here. And once it finds this region, Cas9 searches for the region of homology, which is basically an area of matching base pairs. And once this region is found, a degradation mechanism is activated where endonucleases of the Cas9 induces a double-stranded break. And this break is made upstream of the PAM sequence. So traditionally, the cell repairs the cut by directly joining ends, which is known as the non-homology end joining mechanism, shown on the left here, or it could also use other available DNA to patch the cut and repair. And this is known as homology-directed repair, shown on the right. However, in our case, we could also repair via a base editing technique known as the mismatch repair mechanism. And it induces a single nucleotide conversion, which will be explained in more detail by Cal later. So CRISPR hence is a technology with various applications, but most importantly, it has the potential of being developed into a successful therapeutic method to correct genetic diseases. It would be of great use in editing known and faulty DNA sequences that may cause undesirable traits, such as adult onset hyperlactasia, as mentioned before.
So I'll now provide an overview of our proposed method. We propose the use of a Cas9 cytidine deaminase enzyme fusion to induce a C to U to T conversion and thereby transform an individual that is homozygous recessive for hyperlactasia into a lactase persistent heterozygote. As you can see on the right here, this is our strategy pathway, and various cellular mechanisms will be utilised to mediate this conversion. But before I delve into these, it is important that I first introduce the molecular tool that we are going to use. Our strategy employs the use of a Cas9 protein that has been rendered catalytically dead by an induced mutation. This mutant Cas9, shown here in dark blue, will be also fused to a cytidine deaminase enzyme on the N-terminus, this orange enzyme here, and a uracil DNA glycosylase inhibitor to the C-terminus, this pink protein on the right. These fusions bestow new and specific functions onto the Cas9 to generate a multi-purpose molecular tool. Returning now to our strategy pathway, we can see that each component of the Cas9 has a specific function. Firstly, we'll focus in on the conversion of C to U via the action of the orange cytidine deaminase enzyme. As the name implies, cytidine deaminase enzymes remove an amino group from cytidine nucleosides to convert cytosine bases into uracil bases, as illustrated here in Figure 9. We propose the use of the rat enzyme APOBEC1, as it has been shown in previous studies to have the highest deaminase activity, and also has a preferred sequence substrate of CT or CC, which correlates with our target region shown at the bottom. This deamination will achieve our first aim of converting C to U. A problem arises, however, in that the DNA lesion generated by the conversion will be detected and rectified by the cell through base excision repair, or BER. The presence of a uridine in DNA will instigate base excision repair, in which the enzyme uracil DNA glycosylase, this green enzyme, will excise the uracil so that it can be replaced. The whole success of our method relies on this uracil for later conversion into thymine, and hence we need to bypass this BER mechanism. The most logical action to do this is to introduce a uracil DNA glycosylase inhibitor, or UGI, that will disrupt uridine excision and the ensuing BER pathway. The pink UGI conjugated to our Cas9, shown here, will bind to and inhibit UDG, the screen enzyme, preventing base excision repair, and therefore the uracil will remain in the strand. We now need to mediate the conversion of this uracil to thymine by inducing endogenous mismatch repair. The introduction of a single-stranded break on the strand opposite the uracil is sufficient to initiate mismatch repair, in which the nicked strand is removed and resynthesized. Importantly, the unedited complementary strand is used as the template for resynthesis. So how are we going to introduce this nick? This is where our mutant Cas9 comes into play. Comer and colleagues have recently produced a dCas9 with nickase ability by mutating an aspartic acid and alanine residue, which constitutes just one of the Cas9's two functional endonuclease domains. As Daisy mentioned earlier, the two endonuclease domains usually act in conjunction to elicit double-stranded cleavage, and hence a mutation in just one of these will only introduce a nick in the strand opposite the uracil. This nick strand will be degraded and reformed, and due to the fact that the cell recognises the uracil as a thymine, an adenine will now be paired with the uracil. The final step in the strategy recruits the normal cellular repair mechanisms, as the cell will detect the adenine opposite uracil and demethylate it into thymine. This completes our C to U to T conversion, and we have successfully, albeit hypothetically, produced a lactase persistent heterozygote. To recapitulate all this, we've put together a short animation which runs through our method at the molecular level. Here we have our mutant Cas9 fusion and our CT variant. Cytidine deamination occurs, in which cytosine is converted to uracil. Our pink UGI will inhibit uracil DNA glycosylase, thereby preventing base excision repair, meaning that the uracil remains in the strand. The single functional endonuclease domain of the Cas9 will introduce a single-stranded cleavage on the strand opposite the uracil. This nick strand will then be removed. And resynthesized using the uracil strand as the template. And the cell will demethylate uracil into thymine, thereby producing a lactase persistent heterozygote. This whole method is contingent upon getting the Cas9 complex to the target site and ensuring that the correct cytosine base is converted to thymine. On the left here is our single guide RNA, with our Crispr RNA shown in red, which is specific to the target region. A genome search showed that all but two of the off-target sites for this sgRNA had 3 mismatches towards the 5' end. And we found that this was sufficient to inactivate this sgRNA for those off-target sites. Despite this though, empirical data is still needed to determine the frequency of off-target binding to these other 2 remaining sites that don't have the 3 mismatches. We also are able to utilise the canonical 5' NGG 3' PAM sequence, as you can see here in blue. And this happens to be 12 bases away from our target C. This PAM was chosen specifically as it turns out that 12 bases is an almost ideal distance between the target cytosine and the PAM. This figure on the right here elucidates that this is the case. If we focus in just on the blue line, which corresponds to our CC substrate, we can see that the percentage of deamination was highest when the target C was in position 7 of the protospacer region. If we now look at our test sequence that was used, we can see that the target C that was in position 7 was 12 to 13 bases away from the PAM sequence. From this we can deduce that 12 to 13 bases will be ideal for the purpose that we need. So once in the cell, the single guide RNA and the PAM sequence will guide our complex to the target site. Maya will now explain how we will get this complex into the cell in the first place.
When an adult first begins to experience symptoms of hyperlactasia, it can confirm the presence and location of a mutation in the lactase persistence gene by genotyping the individual. Using our CRISPR-Cas9 method previously detailed, we plan on targeting the small intestine of the patient, as this is where lactose would normally be metabolized. We would be targeting somatic epithelial cells rather than germline cells due to the potential risks, both known and unknown, in editing the DNA of germline cells. And to ensure its efficiency, we would first test our CRISPR-Cas9 technique using small intestinal organoids such as the one shown here. Due to our highly customized CRISPR-Cas9, it would be ideal for our machinery to arrive in the intestinal cells in its protein form. Introducing CRISPR-Cas9 to cells in this form has also been shown to provide rapid gene editing and reduce the number of off-target mutations. We plan on inserting our CRISPR-Cas9 into an adeno-associated viral vector, or AAV, and introducing this vector into the small intestine region either via injection or by ingestion of a capsule. And for a period of a few weeks, AAV would then begin to start infecting the cells of the small intestine. Transduction of the viral vector contents into the host cells would then enable CRISPR-Cas9 to begin modifying these cells. As the number of modified cells increases, so will the amount of functioning lactase enzyme, and eventually these levels will be high enough that the patient can begin to metabolize lactase normally. So AAV was chosen to be our viral vector mostly due to its efficient transduction, with AAV vehicles showing promising execution of both the delivery of CRISPR-Cas9 to human cells as well as resulting gene modifications. This vector also allows for the easy manipulation of the viral genome to include our CRISPR-Cas9 system, and trials have demonstrated that even the entire viral genome can be removed to produce a high-capacity adenoviral vector, also known as HCADV. But even if our AAV vector was not completely gene-deleted, there is good evidence that if you remove early genes E1 and E3 from the viral vector, this limits, if not completely removes, any immune response experienced by the patient. And unlike other potential vectors, such as lentiviruses or retroviruses, AAV does not completely integrate its genetic cargo into the chromosomes of the host cells, and in doing so reduces risks of genotoxicity. So obviously there are many benefits to using AAV as our vector, but there are also a couple of downsides, which Megan will now talk about.
So although CRISPR is an efficient technique in genetically repairing undesirable traits, it may also induce the onset of off-target mutations, presenting a major ethical concern or potential issue of this gene editing technology. The endonuclease enzyme Cas9 is known to produce unexpected DNA indels at the scale of thousands of bases. Off-target mutations that occur in human cells may leave unintended sequences leading to cell death. Therefore, the benefits and risks associated with this CRISPR technology should be carefully considered. Our suggested CRISPR approach, involving the use of cytidine deaminase, may be the most effective and least cytotoxic method, since this enzyme is known to be more efficient in fixing point mutations than the generic CRISPR-Cas9 strategy that cleaves the double strands. Therefore, most of the issues with our suggested strategy are associated with the method of delivery of CRISPR into affected cells. Multicellular cells possess the p53 tumor suppressor gene that plays an important function in the regulation of the cell cycle. Since our method aims to target intestinal cells, tumors are suppressed and prevented from growing if the p53 gene is functioning correctly in these cells. p53 is a natural defense against edits made to the genome by CRISPR technology, where the cell with the modified DNA sequence self-destructs, thereby removing mutated regions and the potential for tumors to form. Therefore, by picking cells that have successfully edited and repaired the damaged gene that initially caused lactose intolerance, we might also be picking cells without a functional p53. Alternatively, the assumption that all cells have functioning p53 genes would also present an issue of our method, since any cell that persists with the modified DNA sequence would eventually be destroyed. Another substantial issue with the delivery of our CRISPR into cells surrounds the packaging of all of the CRISPR components into a single AAV vector. In a single vector, approximately 4.5 kilobases are left for the packaging of all components, including the sgRNA, the dCas9, and control elements. Since our particular strategy would also involve the incorporation of cytidine deaminase into the vector, this would reduce the amount of space even further, presenting a major challenge of this transfection method. However, any other methods of introduction of CRISPR into cells involving plasmids, electroporation, and microinjection are known to be less efficient and more cytotoxic. Therefore, despite the potential issue of packaging AAV vectors, this method of transfection is still believed to be the most efficient in curing lactose intolerance, since these issues are outweighed by its benefits of it being an active, receptor-dependent process that is far more efficient and less cytotoxic than other methods. Thank you.